There are a number of different ways in which alleles can combine to form the different genotypes and phenotypes. But how can geneticists predict which genotype or phenotype is formed, and in what ratios? My name is Alyssa, and in this video we will investigate the different analytical methods through which geneticists can predict these outcomes. The first tool we will examine is a Punnett square, and then we'll look at how to treat test crosses and sex-linked traits within them. We'll then look at gene mapping, and finish off by exploring the Hardy-Weinberg equilibrium. After watching this video, you will be familiar with the different analytical approaches used in genetics that you could see on test day. You will be able to answer questions that test your skills on these approaches by recalling when they can or cannot be used. Punnett squares are a lot of fun and are great visual aids for determining the ratios of genotypes and phenotypes in offspring. The best way to get comfortable with this analytical approach is to make a Punnett square yourself, so let's work through one together. Right now, we will just look at one set of alleles, called a monohybrid cross. So we start by making a 2x2 Punnett square, since we will be analyzing all of the different combinations of one of the two alleles from each parent. We'll use Mendel's purple and white flower example to make this square, where the dominant allele is for purple color and the recessive allele is for white color. Remember that in order for a flower to be purple, it can have either a homozygous dominant genotype or a heterozygous genotype, but if the flower is white, the only genotype it can have is homozygous recessive. Let's say that one parent flower is homozygous dominant. We would then put two capital P's at the top of each column of the Punnett square. Putting the alleles at the top of the column represents the fact that only one allele can be passed down from a parent to an offspring. Let's now cross this homozygous dominant parent with a homozygous recessive parent. We then put the two recessive alleles at the start of each row in the Punnett square, and this represents the alleles that can then be passed down from the second parent. Now our table is set up, and we can begin crossing the alleles of the parents to determine genotypic and phenotypic ratios in their offspring. We start in the top left corner and begin by crossing the dominant trait with the recessive trait. That gives us a heterozygous genotype. We now move down to the second allele from the second parent, and we combine the alleles again to make a heterozygous genotype. Moving to the top right, the combination of the second allele from the first parent and the first allele from the second parent again gives us the combination of a heterozygous genotype. Lastly, we combine the final two alleles to produce the final heterozygous genotype. We can now look at the ratios of genotypes in the offspring, also called the filial or F1 generation. Since this is the first generation after the parent generation, it is denoted F1. Any other generations after F1 will be given higher numbers in sequence. In this monohybrid cross, with both parents being homozygous for either dominant or recessive traits, we can see that 100% of the F1 generation will have a heterozygous genotype. Now let's take a look at the phenotypic ratios for this generation. From this Punnett square, we can see that the ratio of the dominant to the recessive phenotype will also be 100%. Since one parent is homozygous dominant, each offspring will always get a dominant allele, and therefore each offspring will exhibit the dominant trait. But what happens if the parents aren't homozygous? Well, let's have a look. For this Punnett square, we will look at the ratios that result from crossing two heterozygous parents. By taking two heterozygous individuals from the F1 generation, we can create a Punnett square with ratios different from the one before. Again, we are still doing a monohybrid cross, so we only need 4 sections in our Punnett square. Put the alleles of the first parent at the top of each column, and the alleles from the second parent at the beginning of each row. Now we can start crossing them. Starting again at the top left corner, we see that this combination leads to a homozygous dominant genotype. Moving to the second allele from the second parent and the first allele from the first parent, the genotype of the F2 generation is heterozygous. In the top left, crossing a dominant and a recessive allele gives us another heterozygous genotype. And finally, the second allele of both parents will give a homozygous recessive genotype. Now, let's have a look at the ratios from this crossing, starting with the genotypic ratio. Remember that in the F1 generation, the ratio was 100% heterozygous. Looking at the resulting genotypes, we can see that there is a ratio of 1 to 2 to 1, 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Take note that this ratio is only the odds that this genotype will form. With more offspring in a generation, the actual ratio of genotypes will then be closer to this ideal ratio. How does the phenotypic ratio differ from the F1 generation to the F2 generation? From the color coding of the square, we can see that there is a phenotypic ratio of 3 to 1, 3 purple flowers to 1 white flower. Keep this important ratio in mind for test day. We can also use Punnett squares to look at more than one gene. Here we will create a Punnett square looking at 2 different genes, called a dihybrid cross. Think of Mendel's second law here. Inheritance of one gene is independent of the inheritance of another when these genes are unlinked. Therefore, our Punnett square will be larger than before, since we will now be looking at 4 alleles from each parent. The square then has to be a 4 by 4. In this case, we will be examining 2 traits, color and height, where purple and tall are dominant traits, and white and short are recessive traits. For this example, we will be crossing 2 heterozygous genotypes. Because these genes are unlinked, there are 4 possible combinations that would occur during recombination in meiosis. For the first parent, these combinations are again put at the top of each column in the square. The same is done for the second parent, only again the combinations are listed at the beginning of each row. This is the result of the crossing. Quite a complex looking Punnett square, but don't worry, we will discuss what information you should know for test day when it comes to dihybrid crosses. If we look at the genotypic ratio, we can see that there are many different options. Since this is so complex, you are not expected to memorize this pattern for test day. However, it is important to be familiar with the phenotypic ratio of these dihybrid crosses. In these cases, we get a phenotypic ratio of 9 to 3 to 3 to 1. Let's look into what traits are expressed in this ratio. The 9 represents phenotypes that are dominant for both traits, so these flowers would be tall and purple. Both of the 3s represent phenotypes with one dominant and one recessive trait. One 3 represents purple short flowers, and the other represents white and tall flowers. Finally, the 1 of this ratio represents flowers that are recessive for both traits, being white and short. Again, make sure that you can recall this phenotypic ratio for a dihybrid cross in case you need it on test day. Sometimes you don't know the genotype of an individual, but there are ways in which you can figure them out. These cases would occur when an individual expresses the dominant phenotype, like having purple flowers, because you can't be sure if the genotype is homozygous or heterozygous. To determine which it is, you would do a test cross which involves breeding the unknown parent with a homozygous recessive parent. With these test crosses, there are 2 possible scenarios. First, we will look at the resulting Punnett square if the unknown parent has a homozygous genotype. In this case, we can see that all offspring have a heterozygous genotype, but more importantly, we see that all of the offspring will have purple flowers. What if the unknown parent had a heterozygous genotype? In this case, we can see that there are more possibilities for different genotypes. But again, most importantly, we can see that the offspring will have either purple or white flowers, and the ratio of purple to white among the offspring will be 1 to 1. In test crosses, you always breed the unknown parent with an individual expressing the recessive phenotype. That way, you will know if the recessive trait is seen in the offspring, the unknown parent must have a recessive allele and therefore must be heterozygous. Now we will take a look at how we analyze traits that are carried on sex chromosomes. On the MCAT, sex-linked is X-linked, meaning traits are carried on the X chromosome, since Y-linked traits are very rare. Also, unless otherwise noted in the passage, for your test you can safely assume that the X-linked trait will be recessive, which we denote with a lowercase letter written by the letter X representing the sex chromosome. Let's take a look at what this means for both a male and a female. First, remember that females have 2 X chromosomes, and therefore we have 3 possible outcomes for sex-linked traits. The first is that neither chromosome carries the recessive trait, leading to a normal individual. A female can also have 1 chromosome with a recessive allele and is known as a carrier. This individual would not exhibit the trait because both chromosomes must have the recessive allele. Lastly, a female can have both chromosomes with recessive alleles. In this case, her phenotype would express the recessive trait. Now let's take a look at males with an X and a Y chromosome. In this case, there are only 2 possibilities. Either he does not have the recessive allele and is normal, or he has the recessive allele and is affected because there is no normal X chromosome to dominate the recessive one. From this, we can see that males will exhibit sex-linked traits more commonly than females. Let's see what this means in terms of offspring if we cross a carrier female and a normal male. For this Punnett square, we denote the chromosomes as X and Y and represent the affected allele with the lowercase subscript, placing the chromosomes at the top of the columns for the mother and the beginning of the rows for the father. Then we fill in the table. The top right square represents a carrier female, 2 X chromosomes with only 1 affected allele. To the right, we have another female, but in this case, both X chromosomes do not carry the recessive allele, and therefore the female is considered normal. The bottom left is the first male, and since this genotype has only 1 X chromosome, and it carries the recessive allele, individuals with this genotype would be affected by the sex-linked trait. Lastly, a normal male genotype results from the final combination of the X and Y chromosomes. In this example, only the male offspring can be affected by the trait. But if an affected male is crossed with a carrier female, there is the possibility that their female offspring will exhibit the sex-linked trait. Gene mapping involves analyzing which genes are likely to recombine during crossing over in meiosis and explains where genes are located on the chromosome. Gene mapping looks at something called recombination frequency, which is the probability that two alleles on a chromosome will be separated during crossing over. If the two alleles are close to one another on the chromosome, the likelihood that they would separate during crossing over decreases, and if they are beside one another, they are considered linked genes. From this information, we can create a gene map of a chromosome. In this case, we are looking at 3 genes specifically. We can see that A and B are quite close to one another, and therefore their recombination frequency would be closer to 0. There is almost no chance that these genes, since they are so close together, will separate during crossing over. Now we'll compare A and C. These genes are far apart from one another on the chromosome, and therefore the recombination frequency is approaching 50%. This is the maximum value expected because recombination is a random process, where genes either separate during crossing over or they remain on the same chromosome. When analyzing these maps, the convention is that one map unit represents a 1% chance of recombination occurring between genes. Keep this in mind when you are analyzing any gene maps on test day. Lastly, we will explore the Hardy-Weinberg principle. This principle relies on how often alleles appear in a population, called the allele frequency. In other words, this value is the percentage of a specific allele present in the population, for example, the recessive allele that causes blue eyes. There is the possibility that the allele frequency of a population remains constant, and therefore evolution is not occurring. In order for this to occur, 5 criteria must be met. The first is that the population must be large, as this eliminates the possibility of genetic drift. Second, no mutations are occurring that affect the gene pool. Third, mating in this population is random, and individuals are not bred to create specific genotypes. Fourth, new individuals with different alleles are not migrating into the population, and individuals within the population are not leaving. And finally, each gene has the same chance of reproducing, eliminating natural selection. If all of these criteria are met, the population is said to be in Hardy-Weinberg equilibrium. This equilibrium is associated with 2 equations, which we will investigate in detail on the next screen. The first equation we will look at is P plus Q equals 1. This equation describes the frequency of a specific allele. Let's break the equation down so we can understand it. P represents the frequency of the dominant allele, and Q represents the frequency of the recessive allele. Of course, since in the scope of this example there are only dominant and recessive alleles, P plus Q must equal 1, or 100%. The second equation of importance is P squared plus 2PQ plus Q squared equals 1. This equation will tell you the ratio of genotypes and phenotypes in the population. Here, P squared is the homozygous dominant. You can think of it as P times P, which would give you 2 dominant alleles. 2PQ represents the heterozygous genotype, and Q squared is homozygous recessive. In this equation, the sum of P squared plus 2PQ represents the frequency of the dominant phenotype, and Q squared is the frequency of the recessive phenotype. Remember that all the information that you need to solve any MCAT Hardy-Weinberg problem is the value of P, P squared, Q, or Q squared. From there, you can calculate everything else using these 2 equations. These equations may take some getting used to, so let's try out a question that puts them to use. This question is asking you to use both equations to determine the phenotypic ratio of birds with small feet in this population, under the assumption that the population is in Hardy-Weinberg equilibrium. Try this question out on your own, and then we'll work through it together. Let's start answering the question by extracting the known information from the passage. The question states that the dominant allele frequency is 0.4. Therefore, we know that P equals 0.4. The passage also says that we are looking for birds with small feet. The passage states that small feet are the recessive trait, so therefore the question must be asking us to find the percentage of the homozygous recessive genotype in the population. That means that we're going to have to use the second Hardy-Weinberg equation in order to determine Q squared, the frequency of the homozygous recessive phenotype. Now we can begin our calculations. The first step is to calculate the frequency of the recessive allele. We do this using the first equation and by inserting the value 0.4 for P. Solving this equation tells us that the frequency of the recessive allele, Q, is 0.6. Finally, we can calculate the percentage of the population that are homozygous recessive. We take the value just calculated and use it in a portion of the second equation. Remember that Q squared represents the frequency of recessive phenotypes, and so we do not need to use the other terms in the equation. Completing this calculation, we see that Q squared is 0.36. which means that 36% of the population of birds will exhibit the recessive trait of small feet. Great job with that question. We'll finish off the video with a few more practice questions to help you feel confident with these analytical techniques on test day. This question is asking you to think about the circumstances under which we would use a test cross procedure. The answer is B. Remember that in a test cross scenario, we are trying to figure out the genotype of an unknown individual, and in order to do this, we examine the phenotypes of their offspring when the unknown is crossed with a homozygous recessive. Based on the ratio of the phenotypes in the offspring, we can determine the genotype of the unknown individual. This second question is asking you to find the percentage of offspring with a given trait when two heterozygous individuals are crossed. The answer is D, 75%. There are two ways to go about this question. The first is to remember that in a monohybrid cross between two heterozygous parents, the ratio of phenotypes is 3:1, dominant to recessive. This question is asking you to find the percentage of the dominant trait. So by looking at the ratio of 3:1, we know that 75% of the offspring will express the dominant phenotype. We can also use a Punnett square to solve this problem. The parents are heterozygous, so a dominant and recessive allele are put at the top of the columns and at the beginning of the rows. Then we fill in the square. Remember that the dominant trait will be expressed in any genotype that has a dominant allele. And from this square, we can see that 3 of the 4 possible outcomes have a dominant allele, and so 3 of the 4 phenotypes will be brown eyes. Therefore, 75% of the offspring will have brown eyes. Let's try out one last question on this topic. This question is asking you to think about how you would determine the frequency of an allele in a given population. The answer is B, 0.6. To answer this question, we use the Hardy-Weinberg equation p squared plus 2pq plus q squared equals 1. The question stem tells us that 64% of the population has long tails, which means that 64% of the population has the dominant phenotype. Therefore, p squared plus 2pq equals 0.64. We now solve for q squared and find that it equals 0.36. Remember that q is the frequency of the recessive allele, not q squared, so we must take the square root of 0.36, which then gives us the answer of 0.6. Awesome job if you got those questions correct. In this video, we learned a number of new terms and analytical processes used in genetics. First, we learned about the important Punnett square and saw how it helps visualize the ratios within offspring when crossing two known parents. We looked at monohybrid and dihybrid crosses, which look at crossing 1 or 2 genes respectively. And we looked at test crosses, which cross an unknown genotype with a homozygous recessive genotype in order to analyze the phenotypes of the offspring. In this way, we can determine the unknown genotype. We also looked at sex-linked traits and found that for the MCAT, these traits will always be recessive and will always be found on the X chromosome unless otherwise stated in the question passage. We learned about recombination frequency in gene mapping and saw that it was the likelihood of 2 alleles being separated during crossing over. 0% if the genes are close together on the chromosome and are therefore linked, and up to 50% for larger distances between genes. Finally, we examined Hardy-Weinberg equilibrium and became familiar with 2 important equations, one to determine phenotypic and genotypic ratios, and another to determine allele frequencies. Make sure that you are comfortable using Punnett squares and these equations so that you can answer questions relating to this topic with confidence on test day. Thank you for watching this video and good luck studying for your MCAT.